Ahoy, mateys. We're back for our final discussion here on proteins. Be strong, be strong. In this video, we're going to discuss enzymes. So, as we touched on earlier, enzymes are protein catalysts. So, they decrease the activation energy, which increases the rate of reaction. So, obviously, enzymes are proteins. So, everything we said before about proteins still applies here. So, they obviously got to have correct specificity, correct affinity, or high affinity, excuse me. This competition comes into play, saturation comes into play. Needs to be Proteins need to be in a tertiary or quaternary structure to function. Blah blah blah. All that stuff we said about proteins still applies to enzymes, since enzymes are in fact proteins. So, for example, uh, enzymes obviously speed up the rate of the reaction here. So, let's say an enzyme, aka protein, plus a substrate, which is really another name for a ligand. So a ligand binds to a protein, allows it to function properly. So enzyme plus substrate, that'll yield what it's called an enzyme substrate complex. Say that with me. So it's enzyme substrate complex. So if you can see that right there, hopefully it's clear. Enzyme substrate complex. And what that is, it's easier for the substrate to reach its activated state. And obviously a chemical reaction needs to reach its activated state for the reaction to occur, to go to the other side of the hump, if we will, the other side of the hill. Once it reaches the activated state, it goes to the other side of the reaction. So the enzyme plus a substrate yields an enzyme substrate complex, and then that enzyme substrate complex yields an enzyme and the products. So boom, we go enzyme substrate complex, enzyme stays, the products, the result there. So enzymes, they do in a chemical reaction, they do remain and are reusable. So they're often in reversible reactions too. So they could go forward or they could go reverse. But once an enzyme's used in like this specific reaction, it doesn't burn up. It doesn't just disappear into the ether. The enzyme remains. The product is the result, and you could also move back the other way and just keep going back and forth in a reversible reaction there. But enzymes, just remember, they do remain, they are reusable in a reaction. And there is a specific enzyme for a specific ligand, so you just can't use any enzyme with any ligand. There is a very, very specific enzyme that that binds to a specific ligand. So there's lactase, for example, which is helps us break down lactose. So that's lactase is specifically for lactose, and there's other enzymes out there that are specifically for other molecules. But just remember, molecules they do remain, they are reusable in a chemical reaction, usually reversible reactions, and there is a specific enzyme for a specific ligand. So you just can't use any enzyme for any ligand. It has to be that key into the lock, that specific puzzle piece, that specific enzyme puzzle piece binds to the specific substrate or ligand puzzle piece, and boom, it allows the enzyme substrate complex to occur, which yields the products, and then the enzyme obviously doesn't go anywhere. It is reusable. So many diseases in the body are due to either too many or too little enzymes in the body. So every reaction in the body is going to occur without enzymes, but the enzymes obviously make the reactions occur much, much quicker. So if you have too much enzymes, reactions are going to be boom, just happening too quickly. If you don't have enough enzymes, some reactions aren't going to occur in your body, or they're going to occur but not as rapidly as we need them to. So everything needs to be kept within homeostasis, and that proves true for enzymes as well. So many diseases, reminder, in the body are due to too many or too little enzymes. So enzymes are protein catalysts, decrease activation energy, increase rate of reaction. So they're proteins. So there's with the protein ligand binding or enzyme substrate binding, the specificity comes into play, saturation, competition, and affinity come into play. Also, they have to be in the tertiary structures, quaternary structures to function. But this enzyme and substrate form the enzyme substrate complex, which allows the substrate to reach the activated state much quicker, much easier, much simpler, and that yields the products and the enzyme. Enzymes remain, they're reusable, often in reversible reactions. And last but certainly not least, recall that a specific enzyme binds to a specific ligand. So you can't just have a random ligand binding to a random enzyme and vice versa. It has to be specifically catered, that enzyme, to that specific ligand. And it's easy as that, and sadly, that ends our discussion here on proteins. But we're back in the next episode discussing nucleic acid. And boy, am I pumped for it. Let's go.